end, and we'll try and pull those together. But in order for us to be able to kind of communicate those results and various other bits and get you involved, there's also this sign up form. So this is just a Google form, which basically gives us your email address, and it won't be used in any other way other than for just discussing things that come out of this this tutorial. So yeah, take a picture of that if you want. If I can put this up at the end as well, so that you'll have access to that. So that's everything in terms of what we want to kind of as an outline of what we want to do today. To start with, I was going to try and give you a very high level, broad understanding of what synthetic biology is. And I realize it's quite a broad audience here. So this is, for some of you, this might be kind of obvious, but for others, it might not. And I hopefully it will give you a feeling for some of the different perspectives that people have in terms of what synthetic biology is. Now, when we hear the term synthetic biology, and what researchers often mean by the term synthetic biology is very different depending on who you speak to. There are people that see synthetic biology as very much the synthetic part being the kind of the stressed element. And so these are people that try and use new forms of chemistry to create lifelike systems. Juan's keynote on Monday is a really good example of that as kind of almost a bottom-up approach to creating life. There are others that look at synthetic biology in a more, I guess, pragmatic way, and they say that actually we've got the capability to modify living organisms, and those are organisms that we wouldn't necessarily see in nature. So those are synthetic in some sense. They're not artificial necessarily, but they're different from natural biology. And so they take kind of almost like what we would call a top-down approach, actually modifying natural biology or using it in new ways. But I'm going to take for the rest of this talk kind of quite a pragmatic approach and say that, well, synthetic biology is effectively an approach at trying to harness biological capabilities in new ways that we don't typically see in nature. So it's kind of going beyond natural biology, and that's where the synthetic part's there. But it's this kind of harnessing some of the capabilities that we might potentially want to use in different ways. Now, the reason that we might want to engineer biology or modify it in different ways really stems from the fact that it's probably, well, it is the most massively scalable technology that we know about. And it's the most amazing in my view. In biology, you have these nanoscale machines that are able to carry out all sorts of different processes. So we heard about they're able to carry out metabolism to produce energy, components to actually make copies of themselves. They're able to carry out computations. They're able to do all sorts of different things at the molecular scale, at this nanoscale. But through kind of various levels of hierarchy and emergence, those systems can actually be deployed at a planetary scale. So it's a hugely scalable technology. And as an engineer, or a scientist that wants to use these sorts of systems in new ways, this is a sort of technology we'd love to be able to harness. Now, there are a lot of other aspects of biology that we also find really interesting and that I think Susan's talk touched on really, really nicely. The first is that biology has this kind of really weird way of carrying out complex computations and decision-making that we don't always understand how it does it, but it clearly does it. We have this kind of intuition that's doing something kind of interesting and amazing. It was actually seeing this video that got me really interested in biology. Like I said, I come from a computer science background, and seeing this ability for effectively a stack of chemicals to kind of make decisions and guide its movement in this environment, I found absolutely fascinating. Yes. So this is actually an immune cell. It's a human immune cell. It's actually hunting down this little like black thing here. It's actually a bacteria. So this is trying to remove this bacteria. You can imagine if this was in your body, it's trying to remove this, I don't know, infection from your body. So it's an amazing system. And this is all obviously it's a chemical system. There's no electronics. There's no CPU. That's what we would typically design. It's a completely different way of carrying out decision making computation. Another feature that biology is kind of present in biology all over the place is this idea of kind of emergent collective behaviors. And I think one of the most obvious examples of that is through development. So this ability for a single cell to divide and divide again and divide again and develop into a multicellular organism. And those collective of cells to have some really interesting morphological or functional phenotypes that allow it to survive or replicate more effectively. And now I think Susan was kind of alluding to the fact that maybe if we could design these sorts of things, that'd be amazing. And we would love to, but it's really, really difficult because this opens up lots of challenges in terms of how would you make a copy of something unless you've got a separate machine to do it and all these kind of things. So, but this is a kind of capability that we would love to actually be able to harness in new ways. And then the final one I want to highlight is something that I think is really important and which differs from a lot of other types of substrate that we typically engineer. And that's the fact that biology typically can always evolve. It has this inherent capability to adapt and evolve to new conditions or new stresses. And that's something that a lot of, we see it indirectly in systems that we build, but typically we don't design systems with those capabilities, if that makes sense. This is something which is quite unique, I think, to certain biological systems. And this is just an example of, so this is actually a massive agar plate. So you can imagine this is feet by feet, this huge thing. And what they've done is these white areas effectively bacteria, they seeded on boundaries, and then they basically have these stripes of different strengths of antibiotic. So the first stripe at the start, if I bring this back to the beginning, so the first stripe and starts here is a concentration enough to stop the cell growing. They're not able to grow into that normally. But what you'll notice is that there are these evolutionary adaptations you see here that allow some of the cells to mutate in particular ways and actually get around that selective pressure and actually then propagate and grow. And then there's another stripe which is then 10 times the kind of the big starting concentration, which they're then able to overcome. And then there's one at 100 times and 1,000 times. So you have this kind of increasing selection against growth, but the cells are able to adapt and evolve to that particular condition. Now, we don't write computer programs that can do this. We don't make any physical artifacts that are able to do this sort of thing. But it's something that I think, as we start thinking about engineering synthetic forms of biology, we potentially could start to tap into. And I think it's something that really differentiates biology from kind of typically engineered substrates. So those are some of the kind of reasons why we might want to create a synthetic biology, I guess, and actually engineer biological systems in different ways. But how do we even go about harnessing some of that complexity that you see even within a simple cell? So this is a very, very simple mycoplasma cell. So this is the cell that's effectively it's a parasite, and it relies on you providing it with most of the actual resources that it needs to survive. But even though it's not particularly doing that much, the complexity within it is absolutely insane. It's very difficult to know how we would ever go about making something like that, do something that we actually wanted to. But there have been some attempts, and this is kind of where the field is at the moment. Hopefully this will give you an idea of some of the capabilities that we now have. So one of the ways that we can start to kind of break down that complexity is to apply what are typically called kind of engineering type principles to simplify or help our understanding of how we would work with those sorts of systems. And some of those principles are things like standardization and abstraction. So we might have all of these, we might look at it and say, well, all these atoms doing all these complicated things, but actually we can abstract them into certain groups or processes within that cell, which are then easier to understand in terms of interactions. And another really important engineering principle is this idea of decoupling. So although in a cell you have this kind of soup of hugely interacting numbers of parts, actually often that some of those interactions can be very weak and actually could be negligible. And so actually some processes may be quite heavily decoupled from other ones. And that may be important for allowing things to evolve efficiently and do other things. And that's a principle that engineers use a lot. Modularity is something that engineers exploit to be able to build very complex systems because they understand the flows of information and processing. And that's a principle that we can use to start to understand some of these natural biological systems, or even we can implement these features in natural biological systems to make them easier to modify in certain ways. So these are kind of general principles that we can apply to that problem. But the actual field of synthetic biology itself was also founded upon two really key technologies. The first is around our ability to read DNA. So people call kind of DNA the code of life. I'm not sure I like that analogy, but understanding what's in the genetic content of a cell is really important. And so DNA sequencing has allowed us to understand the biodiversity and some of the sort of underlying genetic code that actually underpins some of these processes, which is critical if we want to actually create new forms of biology. But the other really important one is technology that allows us to write DNA. So this is from the 80s, the chemistry that we use nowadays, and it's been progressively refined, I would say. We're still using quite an old technique, but there are some really interesting new techniques for writing bigger and bigger pieces of DNA. And we have examples now where we've been able to effectively resynthesize an entire genome, in this case of a small bacteria and of a yeast cell, where we're not trying to necessarily re-implement or create a new genome from scratch, but we're just trying to see if we can physically or practically build the amount of DNA that's required to create a genome. And so we have those technologies available. But what I think is maybe even more interesting is that some of the technologies that we're beginning to develop allow us to not just create pieces of DNA in a test tube outside of a cell. We have tools within cells themselves that we can start to program or use to modify the genetic code in very specific ways. So I suspect a lot of you will have heard about CRISPR, and CRISPR has been adapted for doing editing of genomes, but also actually inserting large chunks of new DNA or removing large chunks. And so we have a real capability now of not just doing the modification outside of a cell, actually doing it in the cell in vivo, which is really opens up a lot of other opportunities for how we might engineer these systems going forward. So this is kind of the approach that larger areas of synthetic biology take in terms of trying to tame some of that biological complexity. Now, obviously, we've got these technologies, we can write DNA, but what are we going to write if we're not sure? So we can look at what's out there and try and understand some of those processes. We can maybe glean some useful biological components that we can start to put together, and that's precisely what a lot of bioengineers have done. And a lot of the work today around trying to kind of reprogram cells, that I'll call it, has been to take some of those basic components that we understand that have simple functions, like individual genes or regulatory components, and build simple circuits out of them. So you can imagine these circuits are a little bit like little networks where you might have one node is inhibiting another or activating another. And by creating this network with the right topology and the right connection strength, you can generate some really interesting phenotypes. So this top one is an example of an oscillator where you can have populations of cells turning on and off with a certain frequency. And at the bottom, there's been a lot of work around trying to effectively recapitulate very basic combinatorial logic in cells. And the reason for that is that if we have an ability to carry out these basic kind of logical operations, we can use them to help us classify whether a cell is diseased or not, or potentially to make a decision which can be vital in maybe sort of biomanufacturing type applications. But these circuits kind of form a big component of what's been done in the kind of the field today. Now, this isn't just done on a computer or thought about or drawn. These things actually work when you put them into cells. So if I go back to the previous slides, this oscillator I spoke about here, it has one of these interesting properties that one of the components in there is a small molecule that's so small, it can actually diffuse outside of the cell, and potentially other cells can then take up that molecule. And what that does is it effectively couples each individual cell to every other individual cell through the diffusion of that small molecule. And so when you actually build the DNA, can you see my point? Yeah, which implements these various components here, and you put it inside a cell. When the cells grow, nothing looks like it's happening, but when you have enough of those cells, there's this kind of quorum type effect where you have enough of that small molecule being produced, you generate these oscillations effectively in gene expression. And here, these oscillations are actually spatially extended, so you end up with these pulses of activity that you see across the population. Now, I'm an engineer, so you look at this and you think, well, that's kind of cute, but is there actually any use in producing a cell that has a pulse of activity? And there are, because one of the great things about biology is that you can connect what's happening here. So here we're just expressing the fluorescent element that we can then view on a microscope, but we can connect that element to something, the production of, say, a chemical which is maybe able to kill cancer cells. And that's precisely what these researchers did. So here they have cancer cells underneath these kind of sort of strange, like large shaped blobs. And you can see very small little rods that are moving around on top of it. That's a population of bacteria without later. And what they're able to do is to basically allow bacteria to grow. And once they reach a certain population size, they then all produce and release a toxic compound from these cancer cells. And you can see the cancer cells effectively all die. They turn into like spherical blobs, so they're effectively dead. So I think this highlights something where the real power with biology is its ability to connect really interesting physical properties to some of these kind of computations that we can start to generate in living cells. Now, one of the things that my group and other people have also been thinking about is whether the sorts of engineering we do with biology differs in any way from the typical engineering that we might carry out in other areas. And one of the kind of prominent design approaches that's typically used is what's called a design build test cycle. So typically, if you're trying to design something as an engineer, you'll carry out some brainstorming, you'll have a model maybe of how the system works. You might have some physical laws that you know the system's going to adhere to, and you use those to come up with some kind of artifact which hopefully carries out the function that you're interested in. Now, one of the challenges engineers face is that typically that intuition and those models are not perfect. And so there's a gap between the design, how you think the design's going to work, and actually reality. And so you have to actually build a prototype or an actual device itself to then test how that design works. Typically, it won't work perfectly, but through that, you'll learn how to make tweaks to your design that you can then build and test. And so you go through a cycle typically many times in order to produce a working artifact that you want. Now, one of the really interesting things when you compare this kind of cyclic design process to what happens typically in biology is that there's kind of a link to some of the things that biology does when it evolves. So in terms of design, there isn't necessarily I'm not a religious person, but I don't see there being someone actually deciding how a particular genetic piece of information should be modified. But you do have variation, random variation that occurs within an organism. That then leads to expression of that genetic information. So this genotype to phenotype map. And selection is effectively your test there. So you have some kind of removal of phenotype or genotypes which are not functional, or enrichment of phenotypes that are functional or more functional in a specific way. And so one of the things I want to highlight here that we've been thinking about is that when we talk about building or synthetic biology and designing new forms of biological system, we can go about it the kind of the classical way. So we can say, okay, we're going to build our artifact, have a very specific specification, we're going to test it and then see how that works. But because we're building a biological entity, we could, in theory, try to harness the innate ability of that entity to evolve. So rather than us prescribing exactly what the design should be, we could use that inherent evolvability of biology to set up the genetic variations such that it's maybe likely to go in the right direction to produce organisms that have specific features we think might be important to that function, and use clever ways of implementing selection either artificially. So we might select out phenotypes we think are important and only breed those like we do in farming if we want to breed certain phenotypic traits, or potentially implement that selection by having the growth of that organism coupled to some specific function that we want, such that we then just allow evolution to effectively come up with a design that we want. And that's quite a different approach to how we would typically engineer these sorts of systems, but it's something that biology opens up or offers that we potentially could tap into. And one of the nice things of kind of having that more unified approach to engineering design is that you can think then of every single design process as being evolutionary in some way. And this is just an example where we're kind of showing how different, and I'm calling them design approaches. I don't really mean design approaches in like in a classical sense. I just mean they're ways of producing artifacts that have useful properties. You can put them on this diagram here. We have effectively the number of designs that can be sort of assessed at any point in time on the y-axis, and the number of generations you have, or number of cycles you have to go through in order to come up with a feasible design. And what you find is that if you can exploit, for example, directed evolution, then you can have very big population sizes, but you probably can't go through that many generations. But if you can implement selection within your population, so if the population is able to naturally select good designs, then you can obviously scale up the population sizes and also run many more generations without any effect on your system. And some of the traditional design approaches I was speaking about, we typically don't have huge numbers of design cycles, and we can't have millions of potential designs being carried through at every single point. So they typically cluster closer to this kind of bottom left corner. And the reason they work is that we have more knowledge about those systems often. So if we're engineering a bridge, we have certain rules and laws that we can exploit to mean that we don't need to make thousands of different designs. We can single down on those that are functional much more quickly. So I like to use this as a way of thinking about different ways to design systems, and in this case, biology. And I think this thinking about some of these different approaches might be interesting when you start to do the practical exercise itself. To end, I was also going to just highlight one other thing that kind of I spoke a little bit about when we were talking about the design cycle, is this fact that when we're designing biology, we can effectively design the evolutionary process at the same time, which is something we typically don't do in engineering. And so what that really means in kind of a sort of classical sense is that if this was our original design that we've come up with, there's an adaptive landscape around that design that if we allow that system to evolve, it will evolve in certain ways, but we haven't thought about it. So it's going to do whatever it wants to do. But we could design a system where we also think about the ways it can evolve and actually modify the system such that it's maybe more robust. So maybe it's more resilient to mutation. So it might have more redundancy or some other features that actually allow it to be more robust when it's actually being used. Or we might actually design it, like I said, when I went through that example, to evolve in ways that we want it to evolve. So maybe it selects for certain functions that we potentially would want. And the really interesting thing is that there's a lot of technologies now in the biosciences that allow us to start to shape that adaptive landscape in useful ways. So for example, in terms of variation, there are ways that we can robustify the genetics. We can avoid things like repetitive sequences, which typically lead to recombination in biology, to kind of remove that as a failure mode. Or we could actually design targeted recombination. So we could actually put certain sites within our genetic sequence that allow it to alter its structure in different ways to actually cause variation to move in certain directions, which we think are maybe useful. In terms of the actual function of the system itself, we can try and make it more robust by insulating the functionality of the parts we use. This might be through things like feedback control is a mechanism you see in a lot of biological systems. Or we can actually exploit the fact that biology is really sensitive sometimes to small mutations or small changes, which allows it to explore a really diverse phenotypic space by having very, very small or kind of minor changes to the actual genetic underpinnings of the cell. And then finally, in terms of selection, one of the things you see in biology is that if cells are burdened, so if I put something into a cell, I get to produce something that it wouldn't naturally produce, then it would typically find a way of stopping doing that. So if you can reduce burden, the systems become a lot more robust because they don't have those selective pressures to remove those elements that we're inserting. Or what we can also do is potentially come up with really clever ways of coupling the function of our system to the growth of that individual organism. So there you're effectively implementing an artificial selection scheme, but within the biology itself. And so this is just to highlight that there are some technologies now that are starting to become prevalent, and We'll give you a sheet with some other technologies that are available for you to potentially think about, which open up new ways of potentially designing biological systems or engineering. So that's everything I wanted